DS-I Africa. Welcome to the DS-I Africa State of Data Science series. We will be starting shortly. Facilitated by No Innovation. Wednesday, September 30th. On a video chat, a woman smiles.
Good morning and good afternoon. Welcome everyone to the State of Data Science in Africa series, hosted by the National Institutes of Health, the Common Fund. My name is Maggie Dugan and I'm here with my colleagues from No Innovation. We're also known as KI, that's the KI in KaiStorm, the platform you all use to get here today. We are a facilitation team that specializes in running workshops and meetings and events like this to accelerate scientific innovation. Briefly, our agenda today, it's right behind me. We have some welcoming remarks with background on the funding opportunity and a few tips that I will give you for maximizing your experience in this webinar. We'll have an overview of today's session on biomedical informatics and data science in Africa from one of our co-moderators. And we'll have a short presentation from each of our panelists, followed by questions and answers from same panelists, and then a plenary conclusion. It's very straightforward. And immediately after the webinar, we will host some interactive topic discussions. These are, by the way, opt-in, so you're not required to attend, but we hope you will join if you're interested in interacting. It's a chance to continue with the panelists, dive deeper, and to network with others who are interested in the same topic of biomedical informatics and data science in Africa. So today's event is associated with an NIH program on harnessing data science for health discovery and innovation in Africa. I'm going to hand it over briefly to Laura Pavlich. She's the program officer at the National Institutes of Health at the Fogarty International Center, and she can tell you a little bit more about it.
Thank you, Maggie. Just as a reminder for those of you who are, and for those of you who are new to this program, today's event is associated with a new NIH Common Fund program on harnessing data science for health discovery and innovation in Africa. There are 4 different funding opportunities currently open for applications, and you can learn more at bit.ly/NIH-DSI Africa.
bit.ly/NIH-DSI Africa.
Applications are due starting in late November, and we encourage you to visit the KaiStorm platform to learn more about applying to NIH grants and also to meet and network with potential collaborators. Additionally, contact information is listed on KaiStorm and in each funding opportunity for the appropriate NIH scientific contact, and we encourage you to reach out to us with any questions. I'll hand it back over to Maggie to set up the rest of today's session. Thank you.
Terrific. Thank you very much, Laura. Just wanted to mention here that if you are someone who uses social media and you hear something interesting, here's how you can hashtag it. You can use hashtag DSI Africa, hashtag NIH Africa Data, and hashtag No Innovation.
Hashtag KNOW Innovation.
To let people know about what is happening at this session and at any others. So what else? We are recording this session. You will be able to see a recording of this and share that with other colleagues. It will be posted on the NIH YouTube channel, as well as here on the KaiStorm platform. It will have captions also, by the way. What else? If you have any technical questions during this program, you can always go back to KaiStorm and scroll down to the Need Help page. And there you will be able to find someone who can give you help. You can leave a Post-it note, or you can find someone who can give you advice about how to get back here. Questions also. During the panel, you may want to be able to ask some questions, and you'll see at the bottom of your screen there are little clouds for Q&A, and you can write a question there. We invite you, if you see a question that someone else has asked and you think, yeah, I have that question too, or that's a really good question, I want to see the answer to that, please upvote it. That helps us to know which questions have the more energy to them, and we want to make sure that we answer them. I think that's it. We're a large group. We don't know that we can answer every single one, but we'll do our best. So with that, let's get started. I'd like to introduce to you today's panel co-moderator. We have 2 moderators. I'll introduce our first one. Our co-moderator is Geetha Senthil. She is the program director for the Office of Genomic Research Coordination at the National Institutes of Mental Health. I take it to you now.
Thank you. I would like to call on my co-moderator, my partner on this, Dr. Darrell. Welcome everyone joining this virtual session. This panel is on biomedical informatics and data sciences in Africa. As you all know, you probably received an email. This is being held under an initiative by Common Fund NIH called Harnessing Data Science for Health Discovery and Innovation in Africa. And this session in particular will focus on emerging opportunities and challenges on application of data science and informatics technologies in various biomedical domains, and how do you teach those to African data science initiatives and programs. So we will in particular highlight some challenges related to data quality standards when mining large-scale data sets. And we have 5 panelists for this session. They'll be giving short presentations on various data science topics, including open data science in genomics, application of machine learning methods in epidemiology, disease diagnosis, and natural language processing in public health. I'll briefly introduce my co-moderator and myself. You probably got my bios. I'll just go over Dr. Darrell is the branch chief for bioinformatics and computational biosciences branch within the Office of Cyber Infrastructure and Computational Biology at an institute of NIH called National Institute of Allergy and Infectious Diseases. Its acronym is NIAID. And I'm a program director in translational genomics and functional genomics, especially human brain molecular profiling portfolio. And I work within the Office of Genomic Research Coordination at National Institute of Mental Health.
Session goals. 1. Focus on emerging opportunities and challenges in the application of data science and informatics technologies in various biomedical domains. 2. Highlight some key challenges in data mining, data quality, and data standards. 3. How can these methods advance human health in Africa? Data analytics, predictive modeling, including machine learning, simulations.
So I will now turn this over to our first speaker, Dr. Abdoulaye Banire Diallo. He's a professor and director in the Center of Excellence of Research on Orphan Diseases at University of Quebec in Montreal, Canada. He's also co-founder and chief scientist of My Intelligence Machines, startup integrating genomics company. So it's a company that integrates genomics, bioinformatics, and artificial intelligence. Dr. Diallo has a degree in computer science from McGill University. He's also a former research associate at Broad Institute of MIT and Harvard. He has gotten several awards, including the Next Einstein Fellowship Awards. It's one of the top African Scientist Awards. He's currently leading 2 major projects related to precision health for rare and infectious diseases. And he's also serves as an advisor for various science projects and startups in Africa. I will now welcome Dr. Diallo to present his talk.
Thank you, Darrell, for the introductions. And I'm really happy to be here. Thank you for the invitations. My talk will be advocating the need of open genomics data in Africa. How that advocation is going is by presenting example on how we are using genomics data in different areas for delivering services. Starting quickly at the point, for everyone, the introductions know that for the past 20, 30 years, there are a lot of genomics informations that are captured for different layers in the process of biochemical science. And those elements can be grouped by genes, transcript, and other annotations, are now findable, organized, and accessible within different directories, including the NCBI and Ensemble. Okay, we are in, I will just continue from here.
Multiple layers of information and representations. Two graphs show the transition between collecting data into multi-omics, and its following integration, signatures, and therapeutics.
Where we have a lot of data coming from NCBI Ensemble and other consortium that helps scientists throughout the world to design multi-omics analysis, including integrations of genomics with other layer of informations using machine learning, data science to extract biomarkers that are important in therapeutics. So this is a key component towards the discovery of new drugs, and it will lead the potential of bringing new drugs in the market and delivering key services in agriculture and other sectors.
An infographic shows a process. Living cell data informs genome products and interactions. Biologists use this data for experimental measurements, cell variables and DNA sequence. Data scientists make computation models, predictions from novel DNA. Diagnosticians and pharmacogeneticists make assessments and discoveries comparing healthy DNA to mutant DNA.
That is changing the paradigm in how biology is working, and we need to know that all the biologists need to work with data scientists in the coming years, because the data need to be integrated within a systems. And when we take a simplified view of how biologists, data scientists, and medical researchers can work toward genomic medicine, machine learning plays now a central role by turning high throughput measurement into specialized or generalized purpose predictive models. So this is really important to discover and assess new variation within human genomes that could be associated to health or a mutant disease that could lead to a specific disease.
The opportunity. Arrows connect three stages. Genomics 0.1, research on genome. Study the property of genes and genomes. Genomics 1.0, research with genome. Study of diseases using genome. Genomics 2.0, service using genomics at large. Use genomic data for precision human and animal health to maximize agri-food and forestry production.
So that means we are in the era where we can leverage research that is done in studying genes and genomes to include research with the genomes and help our ability to sequence those genomes to understand the structures. A few years later, genomes started to be used to better understand living organisms and their disease states. The development of second and third generation sequencing technology is now moving genomics to a next level, which is to analyze genomic data and really use that to improve human health and deliver key services associated to agri-food and forestry productions.
Principle of personalized, precision, and targeted medicine. A patient group with the same diagnosis and prescription divides into 4 categories. Category 1, the drug is toxic but beneficial. 2, the drug is toxic but not beneficial. 3, drug is not toxic and not beneficial. And 4, drug is not toxic and is beneficial.
It is really important that we figure out, even with this pandemic, one of the key issues on clinical trials for delivering a vaccine is to better segment population that could benefit from a drug and separate those groups from other groups that could be toxic. The drug could be toxic and not beneficial, and deliver the right targets. to identify that one needs to combine the genomics and all the layers informations to better segment how a new drug, a new molecules could affect the pathways related to the immune systems. So this is something that is crucial within the streamline of using genomics and data science within the COVID pandemic crisis.
Explosion of genomic data. A timeline shows historical growth of genome data, starting from 1676 up to 2015.
Well, now we are experiencing a lot of growth of genomic sequence, starting an exponential growth, where we can see that in Africa, we have really low spots where we can access a lot of genomics information. This is information of sequence coming from metagenomics sampling throughout the world.
A world map shows sample sources predominantly from North America and Europe. Only one sample comes from Africa.
And there are a lack of data sampling collected from Africa, which is presented here, even though we have an exponential growth of data collected throughout the world. And how this affects us as a scientist. In my team, we are monitoring HIV mutations and their effects. And with Senthil Hospital in Quebec, we are monitoring how mutations in of HIV in pregnant women can affect structure of proteins that could lead to a resistance. And this is monitored throughout all the sequence strands of the HIV within the pregnant women. We tried to extend such monitoring to study African populations with a team in Senegal, and my former postdoc, who is now professor at the University of Cheikh. And what we realized that we can afford of selecting several strands of HIV throughout the world, coming from open data and from private collaborators' data. But what we can see, it's difficult to model transmissions networks, to study quasi-species, or to either identify functional annotations associated to some of the strands, due to the fact that most of the countries or regions lack a lot of sequencing strands or open virus strands that are sequenced and accessible for analysis.
On a map of Africa, countries are grouped together in 5 different colors. 3 circle diagrams show similar patterns of data in East, North, and Southern Africa.
However, what one can see from the initial collection data, we can see that their patterns between the different regions are really different. We can capture the mutations for the important target mutation sites that we are following on those trackers. But to accurately do that tracking, we need to have more and more data. And that is also affecting how people are tracking now the COVID.
COVID-19 tracking of 8 strains. On a world map, different colored lines connect several worldwide strains of infection from place to place.
In the different region of the world using the next strain tools, one can see that there are a lot of data coming throughout several regions in the world, tracking the mutations of 8 strands that are traveling throughout the world. And we see that we have only small spots coming from Africa. And that cannot help us to better track in the continent, the transmission dynamics and so on. So there are a lot of other questions that could be addressed by the genomics, going from agriculture, livestock. And we call that in my era as genomic informed biovigilance techniques. And this era is a market of $2,000 billion. And I think Africa needs to be part of this new economical market. Let me just finish by showing a quick example how we are using such data in Quebec states now.
How to improve dairy production in Quebec. Regression and classification of profit. Prediction of next lactations. Historical data of 25 years of tests. Influences between factors. Early detection of disease with real-time analysis.
Leading a project where we are improving the dairy sector by helping farmers to predict earlier the replacement of a cow. We are collecting data from the different states, real time, and these data are used. 1.4 million cows are used to design a predictive model that could help predict the lifetime profit of a cow and help a farmer to decide whether 18 months prior he will inseminate or not a cow, depending on parameters. That is a gain of more than $400 million in the dairy sector. So if it is implemented correctly. So I mean, those kind of studies, those kind of services are really important in the development in Africa. However, to do that, we need to have the data that are used in a smart technologies and to have the smart policies that are coming to guide the usage of those data. And also these data need to be under the FAIR principle if we want them to be collaborative, accessible, and if we want the research to be reproducible. Thank you.
Moderator Darrell Hurt.
Fantastic. Thank you so much, Abdoulaye. Very much enjoyed your presentation, and particularly the idea of genomics as a service. I think we'll hear perhaps more about the scaling that I think you're referring to as we go through the rest of our presentations.
Thank you.
There was we've gotten some questions in the chat and we'll try to respond to them, but trying to stay on time. We're going to now move to hear from Elaine and Nsoesie. And Elaine is coming to us from Boston University School of Public Health. And she has a PhD in computational epidemiology, master's in statistics. And her research is focused on digital data and technology to improve health in communities globally. Her work is also focused on addressing bias in digital data and understanding factors influencing disparities in health outcomes, not incomes. She's on the advisory boards of Data Science Africa and Data Science Nigeria. She is also the founder of Rete, which is an initiative focused on providing scientific writing tools and resources to student communities in Africa in order to increase representation in scientific publications. Elaine, we welcome you and look forward to your remarks. Thank you.
Thank you so much for the introduction, Darrell. Today, I'm going to talk to you about digital epidemiology, which is what most of my work is focused on.
Twitter, @ENSOESIE. Email, onelaine@bu.edu.
So if you think about epidemiology as a study of the patterns of disease, injury, and health, and drivers of these patterns in specific populations, then digital epidemiology is basically the same thing, but it tends to focus more on non-traditional data sources, most of the time digital data sources. So an example would be having two kids who have flu-like symptoms, and then the parent trying to figure out whether they actually have the flu or they have COVID. And so they might go to Google to look up information. So this data can be useful at an individual level, but it's also very useful at a population level, because we can look at what is happening in a specific population. And there's been quite a few studies to show that this kind of information tracks with actual reports of disease in different locations.
traditional public health disease surveillance. Points of data are connected in a linear line, starting with public.
So a lot of digital epidemiology at the beginning was focused on trying to see how digital tools can reduce the time between when we have outbreaks and when those outbreaks get reported to the public. So if you think of traditional public health surveillance in this way, where someone gets infected with, it could be a new disease or it could be something we've seen before, and then they go to the hospital, talk to healthcare workers, who then decide this person needs to get tested. And so they get tested, that information is reported to local officials. Then local officials will share this information with the Ministry of Health if it's a disease of importance, let's say Ebola, or it's a novel pathogen like the COVID. And then that information gets sent to the WHO. So before, this used to take quite a lot of time. It could take weeks before that information reaches the WHO. But with advances in technology, we see that this information gets shared a lot faster, and the public can know that things are happening in different sections, parts of the world, a lot sooner than we used to know in the past.
Digital surveillance. Whereas the 5-point chain from public data to world bodies was linear before, now each factor has instant access through digital technology.
And this is what digital epidemiology used to, or digital disease surveillance used to focus on a lot in the beginning. But now the focus has shifted to looking at how this data can be used in surveillance in different populations. So the goal is not to replace traditional surveillance systems, but rather to supplement those systems. So we want to help systems work better. So here are examples of some data sources that I've used in my research. Crowdsourcing, where people opt in and share information about symptoms or disease in their communities. Geoffrey has done a lot of work in this area, so he could be a good person to give some examples. We use a lot of search data from Google, where we look at what people are searching for and how that can inform health in different populations. Social media data, so from places like Twitter, Instagram, can tell us about people's interests, can tell us about people's behavior. So this is a lot of behavioral data. But then there's also remote sensing data, which is not health data, but it is place data. And it can tell us about the environment, and it can tell us how environments can be linked to health outcomes, and think more broadly about the different things around us that influence health. And I think Mustafa is going to mention some of the work that they've done in this space. There's also news data. So an example is Health Map, which is an international or global disease surveillance platform. They mine news reports and put it in one place where you can actually go and see what outbreaks have been reported in your region. And then we have internet forums where people talk about everything from disease to treatments and how treatments are affecting them, that can all tell us about health and disease in different populations. So an estimated 268 million people in Africa used the internet in 2018. That's a lot of people, and they're using the internet for different purposes. But currently, there are more than 200 million active social media users in sub-Saharan Africa. What that means is that people are producing data, whether it's seeking information on health-related issues or sharing information on health-related issues. There's data that's being produced on different platforms, and this data can inform us about health on the continent.
The adoption of these digital platforms for public health surveillance creates complex challenges, requiring the development of solutions specific to the sub-Saharan African context. Nsoesie 2020, Digital Platforms and Non-Communicable Diseases in Sub-Saharan Africa. Lancet Digital Health.
But in order for us to adopt digital platforms for public health surveillance in Africa, we need to think about the challenges that are unique to this context. So what I mean here is that we can't just take what has worked in the West and assume that it's going to work on the African continent as well. We have to think about what are the cultural differences, what are social differences, and how can we incorporate that into the models that we build. So here's an example where we looked at nowcasting influenza-like illness in Cameroon, which basically means we're trying to forecast the present trends in influenza-like illness in Cameroon. And so on one side, you have a plot where you have percent ILI on the y-axis and date on the x-axis.
On the chart, the percent ILU steadily increases in small spikes from 2012 to 2019.
And this shows you our model estimates of influenza-like illness in Cameroon and confidence intervals around our estimates. So our estimates were basically overlapped with the actual data that we got from the Ministry of Health. And to get this, we obtained search data for influenza symptoms, treatments, natural traditional remedies, as well as searches for high-burden diseases such as AIDS, malaria, and tuberculosis, where we were thinking about, like, what would my aunt search for if she had the flu, or what would my grandmother search for if she had access to a computer and wanted to look up symptoms. And what we found was that by focusing on natural traditional remedies, we were actually able to produce models that tracked very well influenza-like illness in Cameroon. So on the other side, you have several different models that we compare. We have support vector machine regression at the top, random forest, multivariable regression models, and then we have autoregressive time series models, where we're basically saying that given that what we've seen in the past on the ILI, can we predict what it will look like in the next time step? And what worked best was support vector machine and random forest in this case. So these are very simple methods that most people learn when they just start doing data science, but we've shown that they're very useful in this particular case if we just want to track how influenza-like illness is, what are the trends in Cameroon. And the main message for this is that context matters. So if we're going to develop models using digital data, we need to think very carefully about context.
2 maps of Africa label countries in relation to the percentages of 2 factors, overweight prevalence and obesity prevalence.
Another area that we've been looking at is chronic diseases. So there's been a lot of conversation around what would happen in the next 20, 30 years in terms of the prevalence of chronic diseases like hypertension, diabetes, and risk factors like obesity and overweight on the African continent. So this is just a map of what this currently looks like, or what it looked like in 2016. You can see that a lot of the higher prevalence are in the North and in Southern Africa. But Africa is the most, is a continent with the highest urbanizing rate, which means that in the next several years, the United Nations actually estimate that by 2050, 56% of people in Africa will live in cities. And so if you start thinking about urban areas globally, you see that these are areas that tend to have higher prevalence of chronic diseases. And there's an expectation that that will also happen on the African continent. So we're starting to think about behavioral data. We know that this data is available from internet platforms, from social media sites. How can we use the data to start thinking about behavior in countries in Africa and track this over time to hopefully give us more information about the prevalence of risk factors?
Information seeking related to behavioral risk factors. Graphs chart 4 different countries and their search terms: Libya, Egypt, South Africa, and Algeria.
So these are the countries that have the highest overweight and obesity prevalence on the continent. So if you look at the countries in North Africa, there's similarity in what people are searching for. So these are search terms. You have yoga, gym, exercise. Then if you look at Egypt, you have gym, breakfast, yoga. So there's a repetition. But then when you go to places like South Africa, you start seeing some differences, like how to exercise, green tea, weight gain. And when I shared this with my colleagues in South Africa, they said there are actually a lot more terms that I should be looking at. At the time, we had about 100 terms. And they said there's a lot more that we should be looking at, and we should be thinking a lot about context, because even though there might be some similarities across countries, different countries think about health differently, and you would see different patterns in what people are searching for. And so when we looked at how the searches correlated with obesity and overweight prevalence, we could see that there were very strong correlations across the continent by just looking at what people were searching for. So the idea is, if we can look at risk factors for chronic diseases, which include things like unhealthy diet, physical inactivity, smoking, alcohol abuse, this kind of data can inform us about what's happening in the continent and can hopefully supplement surveys that sometimes take years for us to see data from. And of course, there are challenges. A shortage of high quality surveillance data is one of them. And I think this actually opens opportunities for thinking about these alternative data sources and how they can be used to supplement situations in which we don't have really good surveillance data. We should also think about cultural influences, as I've mentioned. There's socioeconomic and inequalities and representation. So not everyone is going to be on these platforms because not everyone has the money to be spending time on Facebook or Twitter, or have the resources that they need to be on those platforms. So representation is very important. And then obviously the ethical challenges. So how do we make sure that the products that are coming out of these data sets are not exacerbating existing health inequalities, but are actually helping people? And how do we also use this data in a way that people are informed on how it's being used? So here are some recommendations in terms of addressing these challenges. So digital literacy education, I think, is one that's very important. And we've seen these highlighted during the COVID-19 pandemic as a lot of people buy into misinformation. So I don't think it's something that it's only in Africa, but it's all over the world. So I think digital literacy is very important, educating children as young as possible to understand that everything they see on the internet is not true. Design of appropriate consent system is also important so that people understand how their data is being used. Methods that address a lack of demographic representation is also extremely important. So I want to acknowledge all the people that are working with me on these projects.
Moinina David Senga, Directorate of Science, Technology and Innovation, Sierra Leone. Aristide S. Abba, Ministry of Health, Cameroon. Marshall L. Ndefo Mba, Texas A&M University. Olubasola Oladeji, Tiam Maradi, Dharmesh Tarapur, Kai Jong, and Ziba Cranmer of Boston University. Bukasi Maravate, University of Pretoria.
And thank you so much for your time.
Email, onelaine@bu.edu. Moderator Geetha Senthil.
Thank you, Elaine, for an excellent presentation. It was a fascinating talk, and we learned a lot about digital epidemiology. And now it's time to move to our next speaker, Geoffrey Siwo. Dr. Siwo is he is an assistant research professor in Center for Research Computing and Tech Institute for Global Health, University of Notre Dame, USA. His research interests range from computational biology, network science, artificial intelligence, and nanotechnology. Dr. Siwo was a team leader at IBM Research Africa before he joined Notre Dame. He worked in South Africa, and he also was at IBM TJ Watson Research Center in New York. He was a postdoc at Dartmouth and University of Pennsylvania, received a PhD from University of Notre Dame. He led a lot of publications, patents, international awards, media coverage, startups. So we welcome Dr. Siwo to give his talk on fundamental biology to public health and the science of science with natural language processing. Welcome, Dr. Siwo.
Thank you, Geetha, for the introduction, and thank you, everyone, for coming. So one of the key challenges in biomedical data science, especially in Africa, is lack of data. So today I will discuss how the exponential growth of nucleic acid sequences and biomedical literature, combined with advances in natural language processing, could transform fundamental biology, public health, and even how we do science. And natural language processing is basically teaching computers to read the text, to understand human language.
Opportunity 1. Genome sequences as an opportunity to advance fundamental biology with natural language processing, NLP. A chart displays the steady growth in total bases of GenBank from 2002 up to 2018. An arrow displays the symbolic nature and language-like properties of DNA and RNA.
So I believe that the exponential growth of genome sequence data is a particularly important opportunity for how we do research on fundamental biology. And I think natural language processing has a very important role to play in this. Over the last few decades, there has been a tremendous decrease in the cost of genome sequencing. And this is enabling a very rapid rise of openly available genome sequence data. There are at least two important features of genome sequence data that make them particularly amenable to natural language processing. And one is that genome sequence data, whether DNA or RNA, are very symbolic in nature. So we have an alphabet of A, G, C, T, where A stands for the DNA base adenine. And so it is more than just a letter. It represents a lot of information. And this is also the same of language, because we use language to communicate very complex things about the world. The other important feature about genome sequence data is actually that scientists have used several language-like metaphorical terms when referring to DNA. So we talk of sequence, we talk of a message, we talk of a code, we talk of translation, and even now we talk of editing DNA. And all of these factors make genome sequence data very amenable to processing by natural language processing.
Core question: do naturally occurring DNA sequences across organisms share a grammar?
So one of the key questions that we've been interested in is that, can we learn something very fundamental about the DNA of all organisms on Earth based on their sequences? And we've been exploring how to apply natural language processing to address some of these questions.
Approach: word embedding. Distributed representations of words and phrases and their compositionality.
One particular approach we've used, which is borrowed from recent advances in natural language processing that rely on word embedding. And word embedding basically is a way of developing a vector representation of words in a way that relationship between words in vector space captures their semantic relations. And this approach came out of Google Labs by Mikolov. And so we've been applying this approach to genome sequence data in public databases, including thousands of microbiome genome sequences obtained across the world in different populations, as well as from environmental sequence data. And basically with this data, we're able to train neural networks that begin to capture the relationship between short subsequences of DNA as a means to understand whether there's some hidden grammar within DNA sequences of all organisms. And so I'll just share one of the key important things that we learn. So by applying this approach to DNA without any particular constraints on how we apply the approach, one of the amazing things that we find is that there's a conserved pattern across all DNA sequences of sequenced organisms that basically shows that that trinucleotides, so combinations of 3 DNA letters, have this special relationship where trinucleotides that share the same context across genomes also code for amino acids that emerged at different times in the evolution of life on Earth. So in particular, we find that trinucleotides are codons that code for very ancient amino acids. So these are amino acids that are thought to have formed in the prebiotic world. We find that such amino acids have trinucleotides that share similar context across different genomes. And what this tells us basically is that in the genome sequence of organisms today, there's evidence for the past, and that evidence for the past might actually go up to the beginning of the very early genes and genomes.
Genome sequences as an opportunity to advance public health genomics with NLP.
So next, I'll switch gears to show another example of how natural language processing can enhance public health.
Can we predict whether an animal virus can potentially jump into humans based on the viral genome sequence?
And in particular, we've been interested in understanding whether we can use the DNA sequences or the genome sequences of viruses across various species to predict which of those viruses might emerge into human populations. So a good example is that could we have predicted the emergence of SARS-CoV-2 based on the DNA sequence of the SARS-CoV-2 virus alongside other viruses circulating in the wild? And there are millions of such viruses in different animal species.
Deep viral code, predicting zoonotic viruses from DNA. Challenge data scientists with little biological knowledge to classify zoonotic versus non-zoonotic viruses from genome sequences alone using NLP. Deep learning approach attain the best performance.
So we organized a hackathon that we named the Deep Viral Code Hackathon in collaboration with the Deep Learning Indaba X machine learning meeting in South Africa. And basically we challenged data scientists, many of whom have never worked with biological data sets. We challenged them to consider DNA sequences as text and apply their natural language processing skills to this data set to see whether they could predict whether a virus is zoonotic or non-zoonotic based on the DNA sequence. And one of the key things we learned from this hackathon is that certain deep learning models were actually able to predict very well which virus could be potentially zoonotic versus those that are non-zoonotic.
Opportunity 2. Biomedical literature as an opportunity to accelerate science with NLP. Charts show growth in 3 fields: biomedical articles per year, preprints at PubMed, and preprints hosted by Open Science Framework.
So finally, I want to switch on to another kind of opportunity for biomedical science combined with the natural language processing. And this opportunity lies in the fact that biomedical literature has been growing exponentially. So today there are millions of papers that are published in any one year. We're also seeing an increasing amount of sharing of preprints. And all of this is allowing us to take a peek at science globally in a way that has not been possible before.
A tree graph shows the many facets of learning and information that are all derived from a PubMed query and its XML abstracts.
So we have been particularly interested in applying natural language processing to the field of gene editing using CRISPR-Cas enzymes. And the reason we are interested in this is that we are still in the early days of this technology that could change the way we perform biomedical research. And so we've been looking at publications in PubMed and applying different natural language processing methods to extract different elements from published papers on CRISPR. And the idea is that we want to take the entire CRISPR field and basically map out all the genes, the diseases, the species, as well as the researchers involved in gene editing research as a means of understanding the progress of research in this area, but also as a means of identifying the gaps that need to be filled in this growing area.
Automated mining of global patterns in CRISPR research. A few countries dominate genome editing research. Males dominate, even more so at senior positions. Publications are skewed to certain journals, including paywall journals which may restrict readership. A few authors based in a few institutions dominate. Funding is highly skewed both in terms of the funding agencies involved and the recipients of grants.
And so just to give an example, last year we performed an analysis for Science magazine when they were doing a story on the rise of CRISPR research in China. And basically, by looking at the entire CRISPR literature using natural language processing, we're able to see the contribution of different countries in this emerging technology. Now, I think this is very important, especially for Africa, because we see that there is a huge dominance of both the US and China in CRISPR research. So we need to ask ourselves, what does this mean for the future of this technology in the biomedical field? We also see a lot of disparities in terms of gender. So we see that most of the published studies in CRISPR are led by male PIs. So what does this mean for the development of CRISPR technologies as equitable technologies for human health? And we've also been monitoring the funding mechanisms behind ongoing CRISPR research. And we hope that this can direct future investments in CRISPR research. So finally, I just want to note that for the last 30 or so years, the National Library of Medicine has created wonderful resources that are allowing sharing of vast amounts of genomic data as well as biomedical literature. I believe that to harness the full power of these resources, natural language processing is needed. And I think that because these resources are freely accessible, it's an opportunity for everyone in the world, including Africa, where there might be lack of data sets, to contribute to the advancement of biomedical research. And I want to thank the many people who have contributed in different ways to this work. So one of my students, Kevin Michael at University of Notre Dame, as well as collaborations with IBM Research Africa and funding by the National Science Foundation and an IBM Faculty Award. Thank you.
Moderator Darrell Hurt.
Fantastic, Geoffrey. I really love the innovative application of NLP to genomic data. I think that really captures kind of the spirit of our session today. I'd like to remind and invite the attendees of our seminar today that we have the Q&A feature, and we invite you to continue to add your questions there and even to upvote those questions that you think are most interesting or that need to be addressed. We will have a Q&A session. We are a few minutes behind, but the Q&A session is will be lengthy, and then we'll also have these breakout sessions later where we can continue the Q&A. So there should be plenty of time to ask and respond to these questions, but please do keep sending in your questions through the Q&A feature. Our next speaker is Dina Machuve, and she is coming to us from the Nelson Mandela African Institution of Science and Technology in Arusha, Tanzania. Her research focuses on developing data-driven solutions in agriculture, and she is currently looking at developing poultry disease diagnostic tools using computer vision and bioinformatics methods. She also has a role on various organizations for data science on the continent. So Dina, we welcome you and I look forward to your remarks.
Thank you, Darrell. I'm thankful to be here, and today I will talk about the practical application of machine learning to solve challenges in the poultry sector. And so the application of data science for poultry diseases diagnostics. Tanzania,
Chicken Health Matters,
in East Africa, ranks third among African countries in terms of total livestock numbers. But the country has very low productivity, and this is due to diseases, and we lack data on the sector. It is an important sector because more than 4.6 million households rely on poultry for food, as a source of food, and also income.
Chickens amount to 36 million.
So if we are able to address the challenge on poultry diseases, we'll definitely we have the opportunity to address the second sustainable development goal on zero hunger.
Chicken diseases monitoring now.
Currently, the chicken diseases of Salmonella, Newcastle, and coccidiosis are diagnosed using lab procedures. And I would like to emphasize on Salmonella because it's zoonotic, and really it causes typhoid to humans. And the lab procedures alone take up to 3-4 days to get results. That's normally done using PCR. And also a majority of farmers rely as well on clinical signs. And access to these lab procedures are equally expensive, and they are limitedly available. So it really calls for rapid diagnostics procedures to enable farmers and livestock officers in the field to address the challenges on diagnosing these diseases.
In the livestock sector, data science has potential for early detection and warning of problems.
So this provides an opportunity for data scientists to address these challenges in the livestock sector, where data science tools have the opportunity to provide farmers with tools that they can that will reduce the diagnostics time from 4 days to up to 3 minutes, and enable farmers to make the right decisions, such as isolating the sick chickens to prevent further spread of the disease, and also avoiding the economic loss in a timely manner.
Image data is better than lab data in developing countries.
So we really see the suitable data set being images, because in Africa, we have low levels of literacy and multiple languages. So image data is really a better data set in developing countries. Also, we have the ubiquity of mobile phones. In the continent, we have about 250 million people with smartphones. So mobile phones really provide the opportunity of being a sensor in such venture on diagnostics. So how we apply this on this ongoing project.
A chart shows 4 steps. Fecal images go into a generation of training and test datasets. Along with image processing and agricultural expertise, it gets put into cloud storage. The data is analyzed and then transferred to smartphones for early disease detection.
We have a workflow that has about 4 steps. There's a lot of work to be done on collecting the data on the field before machine learning and data science can come into place. So our task is really to develop a model that can correctly classify images for chicken diseases.
3 images show chickens, the first is healthy, the second has coccidiosis, and the third salmonella.
And here, so we want to, after we have the model, we are able to diagnose and label the right diseases for coccidiosis, salmonella, and whether the chickens are healthy. So this is the data set that we currently are working on. This is the chicken droppings. And this is a sample of how they look like for healthy chickens and for coccidiosis, and as well as salmonella. And we have a target of reaching up to 8,000 images, and currently we are at 2,800 images. So after we have collected the data, we will do the modeling using convolutional neural networks. And convolutional neural networks provide a relatively automated way of extracting different features from the image. And, and this happens on the hidden layers. At every layer, the network discovers a new representation of the input. And at the output, at the output, we are able to make a decision whether the chicken is healthy or diseased. And the label will equally include like what type of disease. And the data set, the raw data set is the chicken droppings images. So we equally envision to deploy the developed model into a mobile application so that farmers as well as livestock officers can have a tool on the field to be able to rapidly diagnose the diseases. it is our vision on this project that we'll be able to have an end-to-end tool for early detection of the three chicken diseases: coccidiosis, salmonella, and Newcastle, and also produce a machine learning data sets to be shared with the larger AI community for further research in the field. And we really equally envision that farmers using this app will have reduced outbreaks of the diseases. Thank you.
Moderator Geetha Senthil.
This was a fascinating talk. It's delightful to learn about how machine learning methods are used to classify images. And I can see how this can be translated to other areas as well. So we will now move to our final speaker. Dr. Mustafa Cissé is a research scientist and head of Google AI Center in Accra, Ghana. Dr. Cissé is his interests are in AI algorithmic development, and he is involved in developing axiological artificial intelligence methods, particularly, and then his research efforts focus on fairness, transparency, reliability of making these methods and data available. Prior to becoming the head of Google's AI Center, Dr. Cissé worked at Facebook Artificial Intelligence in Paris, and he is originally from Senegal, and he did study in mathematics and physics, and received a master's in machine learning and PhD in computer science from University of Marie Curie in Paris, France. We'll now turn this over to Dr. Cissé to talk about improving lives with machine learning. Welcome, Dr. Cissé.
Improving lives with machine learning.
So thank you very much for the introduction and for inviting me. I looked at the previous presentations with a lot of interest. This is a great and timely event in Africa at this moment. So thank you very much. I wanted to talk about what is going on, the various applications of machine learning currently targeted specifically at improving lives, and in particular in Africa. So this is very important because machine learning, and especially computer vision, has made significant progress in the past years. So we went from achieving 26% errors on the standard benchmark in 2011 to having a superhuman performance and recognition benchmarks starting 2016, and we have now even better models. Now, some of this technology can be used to solve some of the most important and pressing challenges of our time, and this has sparked an important wave of interest into various areas of machine learning research. And this is exemplified by one of the signals is the number of papers uploaded on a daily basis on arXiv. So as many as 90 new papers on machine learning every single day. This can be safely called an exponential growth of the field.
2008, USNAE publishes Grand Challenges of the 21st Century.
Now, all this technology that is being developed is being used currently to solve important challenges, as I said before. So in 2008, the National Academy of Engineering of the United States published a list of the grand challenges of the 21st century.
The challenges are: make solar energy affordable, provide energy from fusion, develop carbon sequestration methods, manage the nitrogen cycle, provide access to clean water, restore and improve urban infrastructure, advance health informatics, engineer better medicines, reverse engineer the brain, prevent nuclear terror, secure cyberspace, enhance virtual reality, advance personalized learning, engineer the tools for scientific discovery.
And as you can see, machine learning is being used to, in certain cases, accelerate the discoveries in some of these challenges. in other cases, really as one of the main tools to make progress in these fields. And at Google, for example, we have been looking into all the fields that are listed in red here. The issue with machine learning is that even though it's an important technology, the experts working in this field are not distributed evenly across the different geographic locations of this planet.
A world map shows AI experts working predominantly in North America, Europe, and Asia.
So a few years ago, a company called Element AI sitting in Canada did a survey of the machine learning engineers and researchers to find out where those people are, where do they work? Because this is an important question as it defines to some extent the questions and challenges they decide to address, and also the immediate environmental impact they have, because one cannot detach research questions from the environment in which that research is being done. And their findings are quite telling. From this map, and I'm sure you've seen a similar map in other situations, from this map, we can clearly see that most of the machine learning researchers and engineers in the world are either in North America or in Europe, or to some extent in China, Japan, Australia, or India. The communities of machine learning researchers, as surveyed by the study of Element AI folks, is very scarce within Africa. I want, in this talk, to challenge a little bit this idea, because as scarce as this community is, it is growing extremely fast, and it has a huge potential for impact, as has been discussed before by Dina, for example, whose work in the context of DSA, but also in Tanzania, I would like to commend here, but also by Elaine and Abdoulaye and Geoffrey and all the others. So this is important because we live in a company which will be populated by 2.4 billion people by 2050. And these people will represent 40% of the world's population in working age world's population. So making sure that whatever technology we develop has a positive impact on these people is critical. And this is the work that is being tackled by several groups across the continent, also including the research team I work with at Google.
AI and agriculture. Use AI to improve people and plants' health. Hashtag AI by Africa.
So I wanted to start with this application, which is how do we use AI to improve agriculture? So in many places in Africa, having healthy crops is a matter of food security. A significant part of the population relies on agriculture for income, for their own living, etc. So when it's about humans, when we talk about health for humans, we can have doctors who diagnose humans maybe early in order to take the right treatment. But how about plants? This is an important topic that is not discussed enough. So in Central and East Africa, for example, if you consider cassava, which is an important, one of the most important crops there, so it is plagued often with some diseases. And if we can diagnose these diseases early, using a technology that is affordable, that is accessible to the farmers, we can take action early, and therefore this can prevent significant losses of the crops. So a team of folks.
On a close-up of a plant, diseased leaves are highlighted in blue.
A team of researchers led by Dr. Ernest Mwebaze, who is from Uganda, but who is working in the team at Google AI, developed a technology to perform crop disease diagnosis using cutting-edge research and technology built in the lab in computer vision to build a software that can be deployed on low-end mobile devices so that it can run offline, and that farmers can use in their smartphones to take snapshots and pictures of their crops, the leaves, and then get an instant diagnosis of the plants.
Understand populations.
Another important aspect is how do we use machine learning and computer vision in order to improve our understanding of the populations. Now, this is an important question. Elaine talked about computational epidemiology, and this is a potential source of data for understanding how the populations evolve, where to build new hospitals, which populations are at risk, etc. So one way of doing this is to analyze satellite imagery.
Photographs show 4 cities with different building densities.
So if we can look at the cities, African cities and other parts of the continent from the perspective of satellite images, we can gain a lot of insights into where people live, how many people live there, etc. Some countries, like DRC, for example, have not conducted a census, proper census for 40 years, for example. So it is very important to know how many people live in certain areas and where do they live in order to make certain decisions regarding policy. But African cities are very diverse. As diverse actually as the population of Africa, arguably the most diverse population on the planet as far as the different continents are concerned. And this makes the analysis of satellite imagery more challenging if we want to know where buildings are, etc.
How many people are there? A photograph shows an aerial view of Makoko, Lagos, Nigeria.
So, for example, if you look at this this view of the Makoko, which is a slum on the water in Lagos, Nigeria, can you guess how many people live here? I think this is a challenging question. Or if you look at the high density area in Kampala, can you guess how many people are here? This is also very challenging. Now, all of these...
A digital map shows the roadways of a city.
These cities that you've seen, or these images that you've seen, can be converted into digital maps, just as when you use Google Maps. But when the algorithms that convert the satellite imagery to the maps are not accurate enough, you end up having maps that are pretty empty. So you don't see the buildings on them. If you want to conduct any analysis or any study based on these maps, you will not be using a reliable information because you don't know where the buildings are, even though we saw in the previous images that there are many buildings. In the map, they are lacking. Now,
a satellite map details the missing buildings.
As you can see, the satellite imagery corresponding to the previous map image is here, and there are a lot of buildings and therefore a lot of people.
Buildings detected for addition to map facts. The buildings are highlighted in green.
So we've been working within the lab, a team led by John Quinn, to build improved image segmentation models that can find the different buildings that are in a map, in a satellite image, and then therefore get a better insight into where people live when applied across the continent.
Advanced health informatics.
So one last application I want to talk about, which has huge potential as well to improve people's life on the continent, and which have been developed at Google AI by teams sitting across the world, especially in Mountain View, is how can we use machine learning and computer vision to advance health informatics, in particular to solve diabetic retinopathy. So diabetic retinopathy is the fastest growing cause of blindness. And in particular, if we consider Northern Africa and Sub-Saharan Africa, by 2040, we will have a total of over 100 million people suffering from diabetic retinopathy. Now, the issue with diabetic retinopathy is that it is a disease that we know the current state of medical research allows to solve if it is diagnosed early. The problem is that 45% of the patients suffer vision loss before diagnosis, and this number is reported in a study conducted in India, for example. There is a huge shortage of ophthalmologists in many parts of the world, and Africa is no exception to this. If we can automate the diagnosis of this condition, many people, a significant number of people, will not lose their vision just due to the lack of diagnosis.
How DR is diagnosed: retinal fundus images. Healthy and diseased retina are shown side by side. The diseased has dark splotches, hemorrhages.
Now, how can this be done? One way is just to mimic the way the ophthalmologists do it, which is by looking into retinal fundus images and collecting many images by having them labeled as no diabetic retinopathy or proliferative diabetic retinopathy, and all the spectrum between those two extreme cases by expert ophthalmologists. And once that is done, one can train a deep neural network in order to recognize or in order to predict whether a given image corresponds to a patient who is developing a diabetic retinopathy, who is at risk or not.
Development and validation of a deep learning algorithm for detection of diabetic retinopathy in retinal fundus photographs. The F-score for the algorithm is 0.95, while the median ophthalmologist is 0.91.
Now, what the teams of scientists at Google and other institutions have done is to train such models on large amounts of data. And what they found is that the resulting model can achieve a score in terms of accuracy, which is, to the very least, on par with the average ophthalmologist, but very often which competes favorably with the expert ophthalmologist.
The African Masters in Machine Intelligence.
Now, all these applications that I've talked about are just examples of what we can do. But one thing that is very important is that whether it's data science or machine learning or artificial intelligence, whatever you call that technology, will realize its full potential in positively impact people's life only if the populations have the knowledge to solve their own problems, because they know their challenges, the challenges they face on a daily basis better than anyone. And because of this, we created the African Masters of Machine Intelligence at the African Institute of Mathematical Science, which is the first graduate program in machine intelligence on the continent. We've already graduated close to 100 students in the past 2 years, and those students are working in the continent and elsewhere to solving some of the most pressing challenges that we have. You can find more information about this online. It's an extremely selective program, and we're trying to make it accessible to more people. There is a lot to do, of course, in order to harness data science to improve the health of people and livestock and plants in Africa. There's a long road ahead, but I believe after watching all these presentations that the future is exciting. Thank you very much.
Darrell Hurt.
Wow. That's fantastic, Mustafa. Thank you so much for your kind of survey and of course the examples that you've given. I thought that we've had a great diversity of application of machine learning and data science to different aspects of health and well-being, as well as basic research and biomedical informatics, which is, again, the topic of our session. We've gone a little bit over, and we're going to have our Q&A session. We'll still conclude at 10:30. But for our Q&A session, first of all, I'd like to thank the audience for all of the questions that they've contributed over the course of our discussion here today. And what I'd like to do is maybe just kind of pose some of those questions to our panelists. I think the question that has been upvoted the most is that it's important to have access to clinical data. How can we get access to clinical data for data science in Africa? Any thoughts?
So Geoffrey here, I'll give some thoughts. So I think when it comes to clinical data, there has been, of course, some traditional ways of clinicians collecting data, which is normally through notes. And I think this is an area that there is great opportunity for data science, because with clinical notes, you could digitize them, and once you digitize them, then you could apply approaches like natural language processing to extract information for research. And I've been involved in one kind of that research when I was at IBM Research Africa Lab in South Africa. So we worked with the National Cancer Registry in South Africa to explore how pathology reports, which are basically written notes by a doctor describing a cancer patient's case, and basically we were able to develop natural language processing techniques that could go through these pathology reports and extract more detailed information from those reports, which are just text at the end of the day. So I think importantly, you need collaborations. In our case, we collaborated with the National Cancer Registry, and in any given African country, there are Ministry of Health that perhaps could be very important partners in trying to gain access to the clinical data.
Geoffrey, thank you for that. I agree that natural language processing is an important part of reading the phenotypic or the clinical information, and there's lots of challenges there. I want to make sure that there were also questions asked specifically to Dina, talking about your research and the application. Are you using a CNN architecture tool, or are you developing something, or are you using an existing architecture to develop the model? If so, which one are you using?
Okay, yeah, we are using existing CNN architectures, and we've started the initial training of the images using transfer learning, since our goal is deployment into the field. So we are doing currently training on MobileNet, ExceptionNet, and VGG, and ResNet models. But since our goal is deployment of the model onto mobile phones, so we are going to continue training and deploy using the ExceptionNet. Since we still have little data, we're still training, but those are the transfer learning models that we're using. Yeah.
Fantastic. Geetha, do you have a question that you've identified maybe for our panelists?
Yeah, there was a question for Dina and also for Mustafa. I was wondering if both of you, for Dina, the question is, what challenges have you faced in implementing your solution? And how easily can it be adapted to other livestock? And Mustafa, they're asking, could you please talk about the potential for AI to address public health risk related to environmental pollution in urban areas of Africa? These are for you.
Okay. Mustafa, are you going first?
Thank you, Dina. Feel free to answer first if you would like to.
Yeah. Okay. Yeah. So initially, the challenges that we really face is building trust with farmers. When we do, because we're collecting data at the farms, so it really takes time to build trust with farmers that the solution will directly benefit them. However, when we visit these farms, we are normally accompanied with livestock officers and vets. So we really take time to explain about the research and the expected solution. So to address this challenge, we are preparing ourselves that after we have the tool in our hands, we'll have a lot of workshops and seminars with these farmers to educate them so that the tool is easily adopted. However, we see the livestock officers being the early adopters of this solution.
Thank you. Yeah, I just wanted to offer some thoughts regarding the question that was addressed to me. I think an important point generally when working on this continent to keep in mind is how does the technological solution fit within the broader solution? I think most of the problems that we're tackling here, including in healthcare and in preserving livestock or looking into environmental impacts into health, do not only call for a technological solution. We need to be very humble in this regard and see how the technology that we build, and in this case, machine learning or artificial intelligence, how can it be used either to accelerate the solution that is developed collectively by the different stakeholders, or to enable solutions that would not have been possible otherwise. Now, to be more precise, as far as health risks related to environments are concerned, one avenue I see where machine learning and AI could have a huge impact is in surveillance, for example, because cities grow extremely quickly, slums developed extremely quickly. Sometimes in areas of conflicts, for example, in a matter of 2 weeks, you can see a whole new city with hundreds of thousands of people just appear out of nowhere. We've seen this happen in various places in the continent. So how do we monitor this? How do we very quickly understand the speed at which a settlement is becoming a city in order to provide with all the facilities that will guarantee a healthy environment for the people working there? I think this is very important. It is under researched. It's not the potential for using some of the techniques that I've talked about or that Elaine has talked about before have not been realized yet, that the full potential, but I believe there is a lot to do in this area. There's a lot of other opportunities like monitoring how buildings, where people are building illegal settlements, for example, in areas where this should not happen because it's just dangerous, because it's close to certain mines or it's close to certain factories emitting different kinds of gas or products or whatever. So I think satellite imagery analysis holds a lot of potential in all these areas. But of course, there are other data sources like social media, and Elaine talked about this as well.
Geetha.
Thank you. Thank you so much, Dr. Nsoesie and Dina. We have one question. The question is, have you considered other, apart from DNA in your model, incorporating environmental exposure covariates, and because they are known to also influence the risk, especially for cross-species jumps. And built in that same question is, have we also considered RNA signals in addition to DNA? So that's for Geoffrey.
Geetha, what I'd like to suggest is that we're already over time for our Q&A session. what I'd like to suggest is that we take advantage of the very interactive breakout sessions that are coming in about in less than 10 minutes.
Sure.
And perhaps that was Cecile Vibon can maybe ask that question to Geoffrey directly in that breakout session.
Okay.
There are also many other questions that were posed, and we weren't able to answer all of them, but we will capture them and feel free, of course, to pose them again to the panelists in the breakout sessions. I'd like to offer just a few concluding remarks from my perspective as the moderators, and first of all, to thank again Geetha for serving with me as co-moderator, and especially our panelists. Let me share my screen for just a moment before I turn it back over to Maggie and the KI team. I hope you can see what I've got here, and maybe I took several notes as we went through. I don't have time to recap all the notes, but I wanted to share with you perhaps this one slide, and it kind of comes up from the questions that we've already heard today.
Data science uses the scientific method to turn data into value. Values are conclusions, discoveries, and inventions. Value derived from machine learning is predictive and descriptive models. A Venn diagram shows the combination of communication, statistics, programming, and business makes the perfect data scientist.
That data science, to find the perfect data scientist who has great skills in communication and mathematics and statistics, including probability theory, in programming and IT, as well as business or domain expertise, that's a unicorn. And maybe our panelists are those unicorns, but I think that for most of us, it's going to take a team. It's an interdisciplinary approach. And there's roles for machine learning scientists, data analysts, data engineers, and that this term data science is a very broad term and still not entirely defined. Let's remember that data science is science, but it's an applied science, one that is designed to take data and turn it into value, meaning conclusions, discoveries, inventions, and especially when we use machine learning, predictive and descriptive models, as we've seen today. Models that describe DNA or chicken disease or human health. So again, thanks to our panelists, I invite you to, as we go into this Q&A session, to think about maybe some of these ideas. What are the challenges or barriers to how can we build a community that incorporates all these disciplines? Which applications or techniques have the greatest potential to impact human health and well-being in Africa, and why? So these are questions I invite you to think about as we talk to our panelists in the breakout session, which Maggie will now explain. Thanks, everybody.
Thank you.
I want to thank you very much, Darrell and Geetha, and all of the panelists for a very rich discussion. And to the panelists and the moderators, you may want to go now to the breakout rooms to get ready for the interactive session. And as I mentioned earlier, we will invite all of you to continue the conversation by joining the interactive session, but not yet. I'll show you how to do that in a moment. A few things first. In the chat, you will find a link that goes to an exit poll. And we would really love it if you would take some time to fill it out. I have it here also on the slide.
go.hub.ki/sods_biomedinfo_feedback.
But you should be able to click on the link and give us some feedback about the session. Let us know how it worked for you. It really helps us as we plan future sessions to know how you felt about this one. So please, if you would, I'm going to give you a few moments now. It only takes about 20 seconds to fill it out, only a few questions, but very useful questions for us. So I think it should be this link. If not, Maybe Joe, you could correct the one that's in the chat to make sure that it is that feedback. In the meantime, I'll give you some time to fill that out, please, if you would. In a moment, I will also give you instructions. I'll demonstrate for you how you can join the breakout sessions. We have 3 sessions. They are based on themes that will allow you to have some conversations with each other. And before I do that, let me just give you a little bit of information about what's coming up next. Okay. We have some more sessions on this State of Data Science series. We have approaches to maternal and child health.
October 7th.
We also have data science to fight COVID-19.
October 14th.
And a session on health metrics, measuring, mapping, and monitoring morbidity and mortality at all sorts of levels in Africa.
October 21st.
These are coming up in next week. There's one a week, one on October 7th, one on October 14th, and one on October 21st. I hope that you will join us for those. As well, I just want to reiterate that if you're interested in learning more about the funding opportunities,
bit.ly/NIH-DSI Africa.
Please go to the NIH DS-I Africa site. That's the Common Fund site. Remember those dates, and you can look around, poke around on KaiStorm to learn anything else that you might need to learn.
Application receipt dates, November 24th to December 8th, 2020.
There is an interactive session coming up soon, and I'm just about to tell you how you can get to that. Is everybody ready to learn about that? Have you had a chance to fill out the poll? Then let me share my screen again, and this time I'm going to take you to KaiStorm.
KaiStorm.com.
This is how you got here. In fact, this is the very page that you came to in order to join this session. It is the Biomedical Informatics page. If you scroll down, you will see that automagically a few virtual Post-it notes have appeared. And on each virtual Post-it note, there is a theme. And each theme is a topic that will be discussed in that breakout room. So there's a breakout room on development, therapeutics, and diagnostics. There's one on enhancing existing techniques, and another one on data standards, harmonization, and data quality. So just to let you know, the panelists will be in these rooms, so you can ask them questions. I'll tell you in a moment who's going to start where, but I want you to know that it's not so much a breakout session for the panelist as it is an opportunity to have a discussion with the panelists and other experts from the National Institutes of Health around this subject matter. But to begin with, Doctors Abdoulaye Banire Diallo and Dr. Geoffrey Siwo, and also one of our co-moderators, Dr. Geetha Senthil, will be in data standards, this one here. And Doctors Elaine, Nsoesie, and Moustapha Cissé will be in enhancing existing techniques. And Dr. Dina Machuve and also Dr. Darrell, our co-moderator, will be in development, therapeutics, and diagnostics. And of course, if you get lost or you're confused, or you don't understand what to do, you can always go into this one.
Tech Help Desk.
The way it works is you see each one of these has its own Zoom room. So before we finish today, when you leave this Zoom room, when you leave this webinar, you can go back to this page. It should be on your browser because I think you used it to get here, but if not, you want to go to the Wednesday, 30th September Biomedical Informatics page, and you'll see it, and then you click through the Zoom room, and that's how you can join the conversation. What's important to know is if you go to a conversation and you want to leave and go to another one because you're curious about that topic too, it's perfectly fine. Those panelists will be in those sessions, at least at the beginning, maybe for the first 10 or 15 minutes. They may stay for the whole time, but they might also be interested in another topic and leave too. So if you want to talk to one of those panelists in particular, please go to the session that I mentioned earlier. You can leave and go between the different Zoom rooms as you need. I think with that, I'm ready to say thank you to, again, the panelists and the moderators, to all of you for being here with us. Those sessions are going to last about 30 minutes. At the end of the 30 minutes, you can continue by going into our network lounge. If you have any questions, go to the help. Thank you for the exit poll. If you haven't done it yet, it's still in the chat. Please do it. Hope to see you at future sessions in the weeks coming and in the breakout sessions coming next. Thank you, everybody. Goodbye.
Thank you for participating in this DS-I Africa State of Data Science session. Visit Chi Storm for upcoming sessions.